So, second lecture, we spent Tuesday morning talking about the action potential, the membrane potential, the electrics, how it moves along the axon, and so on. What we're going to address today is what happens to that action potential when it gets to the end of an axon. What happens at the synapse? And this is an image of a neuromuscular junction, deliberately so because the neuromuscular junction is the synapse about which we know the most. So even though there are lots of different kinds of synapses and lots of different flavours to them, most of what I will be saying this afternoon is generalised from the neuromuscular junction. You'll find synapses that are different to the neuromuscular junction, but people tend to think, oh look, and it's different to the neuromuscular junction. The NMJ is the kind of standard, this is what a synapse looks like, and this is what it does. Let's start by looking at the case of Otto Lowy. Otto Lowy won the Nobel Prize for Physiology and Medicine for his contributions in 1936. And at the time that Otto was working, there was a big controversy about what happened at synapses. It was known that synapses existed, and the electrical activity of axons was well known by that time. But there was a debate about what happened to that information when it got to the synapse. And there were two camps. There was a camp that said, well, it's transmitted across the synapse as an electrical impulse. And there was the camp that said, well, it's converted to a chemical signal and transmitted across as a chemical signal. There was good evidence, really, on both sides of that debate. And just to pick a couple of examples, there's a delay as information crosses synapses, which kind of speaks to it takes time to turn an electrical impulse into a chemical impulse and then back to an electrical impulse again. And on the other side of the coin, well, it's very energy inefficient to do that. If you've got an electrical signal, why turn it to something that's chemical only to turn it back to something that's electrical? What's the business there? What's the advantage? So in that environment and that debate, Otto Lowy had a dream. And he was very excited about his dream. He woke up in the middle of the night having just had his dream, and he realised that this was the dream that was the experimental design that was going to allow him to prove whether or not synaptic transmission was electrical or chemical. So he scribbled the results, or scribbled his idea for the experiment down on a scrap of paper and contentedly fell back to sleep. When he got up the next morning, he looked at the paper and was horrified to realise that he couldn't actually decipher what he'd written down in the middle of the night. So he had a bad day, but luckily, the very next night, actually on Easter Sunday, he had the same dream. Lucky Otto. And this time, he didn't just wake up and write the dream down. He woke up and he ran off to his laboratory to do the experiment before he forgot what it was all about. And in so doing, demonstrated whether or not synapses were chemical or electrical and won himself a Nobel Prize. So let's look at the experiment that he did. He created a preparation of a frog heart in a bath of artificial extracellular fluid, and he took out the vagus nerve and put it on an electrical stimulator. And when you take a frog heart out of a frog and put it into physiological saline, it obligingly carries on contracting for you. When he stimulated the vagus, he found that the heart slowed down. And that wasn't very exciting. It had been known that stimulation of the vagus nerve slowed the heart for a good long time. But what Otto did then was he took another frog heart, put it into a similar bath of physiological saline, and he arranged for the outflow from this bath to flow into this bath. And then he stimulated the vagus to this heart, and lo and behold, the heart slowed down with vagal stimulation. But what he also observed with a time lag was that the second heart also slowed down with vagal stimulation. So stimulating the vagus of the first heart was affecting both hearts. And he concluded from that there must be a chemical which was getting into this perfusate in the first chamber and getting into the second chamber, which was affecting the heart in the second chamber. Therefore, synaptic transmission must be chemical. And because the chemical came from the vagus, and because he was German, he called it vagus stuff. Stuff that comes from the vagus. Germans are like that. They're very sort of logical people. And that is the chemical that we now know as acetylcholine. But it was the first neurotransmitter to be discovered, and not only was it discovered as a neurotransmitter, but it laid to rest, or at least Otto Lowy thought, laid to rest this controversy about whether or not synapses are chemical or electrical. The really interesting thing is that today we know that there are both chemical and electrical synapses. So it's one of those happy arguments where both sides are right. The electrical synapses are gap junctions, which are where the cell membranes of two different cells, this cell at the top and the cell at the bottom, are closely adherent to each other, such that there are pores which allow electrical conduction directly across the cell membrane. Now, they are relatively rare in higher mammals such as ourselves, although we do have them. They are much more common in invertebrates and especially in insects and in other sort of more simple animals. So the features of this neuronal gap junction is that it allows passive electrical conduction. A change in membrane potential in this membrane or in this cell directly influences the membrane potential of the other cell. They can be bidirectional or they can be gated, so they can effectively act like a piece of wire or a diode. They can allow change in one or both directions. There is no synaptic delay between the two sides, but oddly they only really respond to prolonged stimuli. And that makes sense. If any of you have done any electronics, you will see, looking at this in an electronic sense, that it looks a lot like a capacitor. Yeah? Two resistive plates between two conductances that are closely opposed to each other. And you would expect that as this is acting as a capacitor, it would respond better to a prolonged stimulation, a low-frequency signal, than to a high-frequency signal. And so they will tend to block high-frequency signals, but respond to low-frequency signals, signals in the milliseconds range. They can, in some circumstances, be inhibitory, and they tend to exist in areas where you want a group of excitable tissues to work as a single unit, as what we call a syncytium. So where we tend to see them, as well as between neurons, is between smooth muscle cells, between cardiac muscle fibers, areas where you want a direct conduction between those tissues. Chemical synapses are the synapses that we tend to be much, much more familiar with. And in chemical synapses, we have a presynaptic electrical change. An action potential arrives at the presynaptic knob and depolarizes it. And that causes secretion of a chemical transmitter that we call a neurotransmitter. And that alters the function of the postsynaptic neuron in ways that can be either directly electrical and involve the depolarization of that neuron, or at least the relative hyper or hypopolarization. They can involve a second messenger, a chemical inside the postsynaptic neuron that causes chemical changes within the neuron, or they can cause metabolic changes in that postsynaptic function. And again, because of the neuromuscular junction, we tend to think of the postsynaptic function being an electrical change, but it isn't always. Sometimes it's a chemical or a metabolic change. So what I'm going to do now is I'm going to go through the chemical synapse in a bit more detail and really give you pointers as to some of the processes that you need to know about in terms of how it works. Because when you look at it like this, it all seems very easy. But there are lots and lots of different processes that take place along that way which can alter the physiology of what's happening. So here's a fairly horrendous diagram of all the things that happen when a chemical synapse is stimulated. And in fact, this is an overcomplicated diagram in that it's cheating and it's trying to show you the functions of several different chemicals at the same time. So that here we've got a receptor binding and a postsynaptic electrical change. Here we've got second messengers and a postsynaptic chemical change all going on at once. So it's not quite that complicated, but there are lots of different options for what can go on. So we're going to go through that diagram and just point out some of the major steps in what's happening as a synapse transmits. The first thing then is that there is presynaptic depolarization. The presynaptic membrane depolarizes and undergoes an action potential. And that action potential opens voltage-gated channels in this membrane, but this time they're not voltage-gated sodium channels as they are in the axon, they are voltage-gated calcium channels. So we get calcium channels opening all around here in the presynaptic membrane. And those calcium channels allow an influx of calcium into the presynaptic knob. Calcium is a very interesting cation in the sense that when it gets into cells, it tends to do clever things within the cell. So that you'll recall that muscular contraction has a lot to do with calcium, and also synaptic chemical release has a lot to do with calcium. It's interesting to note that sodium and potassium are not required for synaptic transmission. If you block the influx of sodium in the axon here, then you will completely block transmission of the action potential. In fact, that's how local anesthetics work. They're sodium channel blockers. They block axonal conduction by stopping that sodium from going across the membrane. But if you block sodium channels in this part of the membrane, that will have no effect. Once the action potential gets to the synaptic knob, it's all about calcium, and sodium and potassium don't have a role in that. So we've got an influx of calcium. And that influx of calcium, first of all, primes the vesicles. The presynaptic knob is full of many, many, many vesicles. And those vesicles are spherical, bubble-like structures of cell membrane which contain the chemical which is the neurotransmitter. And activation means that those vesicles are dislodged from the cytoskeleton. They're sitting within the synaptic knob, happily attached to the cytoskeleton, not going anywhere, and then the calcium knocks them off the cytoskeleton and allows them to move around. The vesicles dock with the cell membrane, and they do that by associating with the intracellular portion of membrane-bound proteins. So there are proteins within the cell membrane that are sending out attractors into the synaptic knob, and when the vesicles get into those, they're attracted and they dock onto the cell membrane. And because of this vesicle system, we get neurotransmitter release in packets or quanta. That quantal release is very easy to demonstrate. If you have a synapse like that, and you put two stimulating electrodes, one here presynaptically and one postsynaptically, and you measure the postsynaptic membrane potential with some measurement electrodes here, what you'll find if you inject current into the postsynaptic area, the postsynaptic neuron, or effectively alter the voltage, is that As you add current, the voltage changes, and the change is linear like that. The more current you add, the more voltage change you get. Not a surprise. If you inject current presynaptically, you get a change in voltage which goes stepwise like this. And that stepwise change in the voltage with different amounts of current is because of the vesicles which are being dislodged in the presynaptic membrane. You can dislodge 2 vesicles or 3 vesicles or 24 vesicles, but you can't dislodge 8.5 vesicles. So each of those vesicles carries a packet of chemical which alters the postsynaptic membrane potential by a certain amount. It goes in jumps. And in fact, it's possible to measure that so sensitively that you can do experiments whereby you can calculate the amount of neurotransmitter that's in a vesicle by looking at the size of this quantal change in voltage. You can say, well, this change in voltage is so big, and we know that if we inject so many molecules of neurotransmitter directly into here, we get a change that's this big. Therefore, each vesicle must contain this many molecules of neurotransmitter. It's a very stable, very sensitive system. The vesicles dock with the presynaptic membrane by the association of intracellular membrane-bound proteins, and there is a quantal effect. So it's a finite number of vesicles. When the vesicles dock with the membrane, they are effectively burst and unzipped, and they empty their neurotransmitter into the synaptic cleft. Now, the interaction between the membrane of the vesicle, the membrane of the cell, the proteins in the vesicle, the proteins in the cell membrane is extremely complicated. Don't go there. It's really, really molecular and complicated. Lots of different things going on, lots of regulations. Basically, all you need to remember is that it's energy dependent, it requires ATP, and it's calcium dependent, it requires calcium. And if you've got those vesicles activated in the presynaptic knob and you've got calcium and ATP there, then they will dump their neurotransmitter into the synaptic cleft. So this is a very tastefully coloured electron micrograph of a synapse, or at least the presynaptic knob of a synapse. This is the synaptic knob, and these are the vesicles all bound into their cytoskeleton. So that when the action potential comes along and the calcium comes in, they're freed up, they move towards the membrane, and they are released into that synaptic cleft. And if you look at that cleft, this is the postsynaptic membrane, it's not very thick. So diffusion across that cleft is extremely rapid. Let's look at the life cycle of a vesicle then. The vesicles are manufactured in the neuronal cell body from smooth endoplasmic reticulum. They break off and they are transported down the axon to the synaptic knob. Once at the synaptic knob, they get filled with neurotransmitter, and then they get bound to the cytoskeleton of the synaptic knob, and that's where they stay until something exciting happens. Once they bind with the cell membrane and release their neurotransmitter, they're then cleft away from the cell membrane, and they bind together in these larger vesicles which look like smooth endoplasmic reticulum. And then they go round and round this cycle. They get refilled with neurotransmitter and round they go. And if you think of it, that's quite useful, because if they just bound with the membrane and then nothing happened, the synaptic knob would gradually get larger and larger and larger and larger as it got more and more cell membrane from all these vesicles. So this kind of vesicular cycle is a sensible way to deal with the vesicles. Eventually the vesicles get old and tired, and then they're transported all the way back down the axon to the cell body where they're destroyed and metabolized. So if you imagine in some of the long neurons of the body, these little organelles are made a good metre or metre and a half away from where they're actually useful, and then when they've finished being useful, they go back a metre and a half back to the place where they're destroyed. It's an extremely well-regulated but energy requiring process. The capability of the axon to transport vesicles in both directions at once is also worth talking about, but again it's a very complex process and one that isn't really fully understood. This circle of vesicle release takes, in my mind, a surprisingly long time. From a vesicle detaching to the cytoskeleton, it takes about 35 seconds for that vesicle to go right round the loop and to be reattached to the cytoskeleton. And in cellular electrophysiological terms, that's a huge amount of time. But that speaks to the small fraction of vesicles that are released with each action potential. You don't get an action potential and every vesicle in the knob goes running for the synaptic cleft. Okay? You get an action potential and a small proportion of these vesicles release their neurotransmitter into the synaptic cleft and are then recycled. And you need to have enough vesicles to last you for at least half a minute or a minute of neurological function before you can start to recycle the first ones. Neurotransmitter has been released into the synaptic cleft. We'll talk about what it does to the postsynaptic neuron in a minute. First of all, we need to talk about what happens to the neurotransmitter afterwards and also the neurotransmitter's presynaptic effects. Neurotransmitters are either hydrolyzed within the synaptic cleft and inactivated, or they are taken back up into the presynaptic knob. And which of those two things happens depends really on the chemical nature of the neurotransmitter. If it's a garden variety neurotransmitter that's really easy to manufacture, like acetylcholine, then it tends to be hydrolyzed. The choline is taken back up into the synaptic cleft, and the acetyl, well, hey, you can get acetyl units from anywhere, so that's not a big deal. We'll just grab one from here and we'll use it next time. If you've got a more complex molecule, like an amino acid, or even a number of amino acids strung together into a peptide, well, that took quite a lot of energy to make, so we're not going to break that down after just one use. We're going to suck that back up into the presynaptic membrane and use it again. So these things tend to get cycled as an energy-efficient usage of the molecules. As well as having receptors for these neurotransmitters on the postsynaptic membrane, there are often receptors on the presynaptic membrane. And those presynaptic receptors have effects on the presynaptic knob. So that, for example, in the neuromuscular junction, there are presynaptic acetylcholine receptors, which are calcium channels, which allow even more calcium to flood into the synaptic cleft to amplify the chemical changes which are going on. So that the amount of neurotransmitter release can alter the release of more neurotransmitter by that synaptic cleft at the time. Usually it amplifies it, sometimes it diminishes it, sometimes it reduces the time course of the release. Those are all presynaptic effects. Let's look now at the postsynaptic effects. Three sorts of changes that we can get. We can get conductance changes. The porosity of this postsynaptic membrane to a population of ions changes, and that leads to a change of membrane potential in this postsynaptic membrane. And just like the rest of the presynaptic neuron, if this postsynaptic membrane reaches threshold, then an action potential continues along the way. Changes that we get there, and we'll see this in more detail in a couple of lectures' time, so I'm not going to allude to it much today, but the changes that we get in this postsynaptic membrane can either be excitatory or inhibitory. Yeah, we can get excitatory postsynaptic potentials, membrane potential changes in the postsynaptic membrane, or inhibitory postsynaptic potentials. And usually, although not always, it's the case that excitatory postsynaptic potentials are due to the opening of chemically gated sodium channels in this postjunctional membrane. So that sodium floods into the neuron and the membrane potential is reduced, is made less negative. Sometimes these are inhibitory, and those are usually, though not always, chloride channels. And so we make the membrane more porous to chloride, and we pull the membrane potential towards the equilibrium potential for chloride. So we make it more negative or hyperpolarize it. And it's all kind of pushing it towards threshold, pulling it away from threshold with these chemically gated channels, so that when the thing reaches threshold, if it does reach threshold, the voltage gated channels will cause an action potential which will move on down that neuron. So those are the conductance changes. And a synapse that works through conductance changes, or a synapse that works through conductance changes, or a neurotransmitter that is causing conductance changes, we call ionotrophic. It's affecting the ion conductance of the postsynaptic membrane. We can also get second messenger systems whereby the neurotransmitter attaches to a receptor on the postjunctional membrane and doesn't cause an electrical change, but causes second messengers, usually G proteins, to be released into the postjunctional membrane, and those can have all sorts of effects within the postsynaptic cell. They can either affect the conductance pathways, so they can amplify or diminish the effects of the ionotrophic activity, or they can cause changes in protein expression, which can go on for some little time. And of course, changes in protein expression can hugely influence the function of these neurons. If you want to look at a really good example of a complicated receptor that's got loads of active sites and is ionotrophic and metabotrophic, have a look at the GABA receptor. The GABA receptor is an important central nervous system neuroreceptor, and as receptors go, we know an awful lot about its function, a lot about the ligand sites, a lot about how different changes interact with the GABA receptor to give us different effects. So it's a really good example of comparing ionotrophic and metabotrophic effects. Just going to talk briefly about the classification of neurotransmitters. They tend to be put into three groups. The first group are the low molecular weight neurotransmitters, and they share very little in common except that they are low molecular weight. And if you think about it, these are molecules that we want to diffuse across a space, the synaptic cleft. So it's kind of handy for their function if they are low molecular weight, because then they're going to diffuse more rapidly. And most of the major neurotransmitters are in this low molecular weight category. Acetylcholine, glycine and GABA and glutamate in the central nervous system, the catecholamines, adrenaline and noradrenaline, dopamine, serotonin. Most of the major neurotransmitters that you'll have heard of are in that low molecular weight molecule category. The other major category are the neuroactive peptides. And these things tend to be chains, small chains of amino acids. You couldn't quite call them proteins, but they're much larger molecules than the low molecular weight molecules. And they also tend to have a little bit of a cloudy grey function, in the sense that if you look at all of these molecules, they're pretty much all classic neurotransmitters. They all work at a synapse. Except, hang on a minute, what about adrenaline? Adrenaline is a catecholamine, but it gets into the blood and it circulates, so it has some functions that are not directly related to a synapse. When you get to neuroactive peptides, you tend to think, well, vasopressin, oxytocin, I've seen those as hormones. They've been in the blood, they're going around the place, they're doing things at distant sites. But we also find them as neurotransmitters. And if I were to put another group of molecules along here out the other way to complete this picture, they would be the hormones. things which you don't tend to find at synapses, but you do find that they get sent into the blood to distant sites. So there's a kind of a grey area between a classical neurotransmitter and a classical hormone. And that's kind of where most of these neuroactive peptides sit. But if you think about it, the function of a neurotransmitter and the function of a hormone, they're not really that different from each other. It's just that they are or are not associated with a synapse. And finally, we've got some things that we call unconventional transmitters. Things like nitric oxide and ATP, which do, in some circumstances, function as neurotransmitters, but they're not the usual run-of-the-mill kind of thing. Two main sorts of functions of neurotransmitters: ionotropic neurotransmitters stimulating ion channels, metabotropic neurotransmitters mediating second messenger systems within the postjunctional cell. Let's have a look at an ionotropic receptor. This is a typical ionotropic receptor. It's a very large protein molecule, usually composed of several subunits. It sits across the postjunctional membrane, and it acts as an ion channel. It has an active site or a binding site for the neurotransmitter, and it has a gate which either opens or closes to allow certain sorts of ions to diffuse across the receptor. If it's excitatory, i.e. if stimulating it leads to excitatory postsynaptic potentials, it tends to be a cation channel receptor. Usually, though not always, a sodium channel receptor. And if it's inhibitory, i.e. stimulating it leads to the generation of inhibitory postsynaptic potentials, it tends to be an anion channel, usually but not always chloride. The characteristics in terms of function of the ionotropic receptor are that it functions instantaneously. When a ligand binds to this active site, it changes state and the gate opens instantaneously. And its function lasts for a few tens of milliseconds. So it's a rapid, short-term thing. If we look at a metabotropic receptor, there are similarities and there are differences. Similarities: it's a protein. It's embedded in the postjunctional cell membrane, and it crosses that membrane. It's a transmembrane protein. But in terms of differences, another similarity, it has a binding site for its neurotransmitter outside the cell. But it doesn't have that classic ion channel of the ionotropic receptor. So when it's stimulated, there tend to be G-protein interactions down within the postjunctional cell, which can alter the function of ion channels, and they will tend to make the ion channels more or less attractive for their own ligands to bind to. So you'll either get increased binding or decreased binding to the ion channel ionotropic receptor, or sometimes they initiate second messenger systems which can mobilize intracellular calcium, and intracellular calcium, when it's mobilized, can do all sorts of interesting things, or can regulate gene expression and therefore protein expression within that postjunctional cell. Typically, the onset of their activity takes a few hundreds of milliseconds, and it can last for hours. So these are not the rapid-firing, responsive things that we think of when we're thinking about ionotropic receptors. They are taking much longer to activate, and once they do activate, their effects can last for a good long time within the cell. Finally then, I want to talk about synaptic blockade, ways of preventing the transmission of information across a synapse. Classically, the textbooks will tell you that there are 5 different categories of synaptic blockade. Don't get excited about dual block, which is always the final category. It's a bit of a cheat, and you don't really need to worry about it, apart from knowing that it's there, which is why there are 5. And note, in this diagram, all the examples are taken from the neuromuscular junction, which tells you how much more we know about the neuromuscular junction compared to the other synapses of the body. So the first group of blocking molecules are presynaptic anti-depolarizing. i.e., they get onto the presynaptic membrane and they prevent it from depolarizing. It can't depolarize, its calcium channels can't open, it can't initiate that chemical transmission, and so the information transfer is blocked. The classic presynaptic antidepolarizing toxin is tetrodotoxin, the most potent toxin that there is. And the one where if you write to the New Zealand government to get a license to import it, just all the bells go off. People that you never know existed come knocking on your lab door saying, why do you want that molecule then? Very similar is botulinum toxin, the active ingredient of botulism. The aminoglycosides are a group of antibiotics which famously have neurological side effects. Not usually a good thing, but common. Not surprisingly, depleted calcium, because if you deplete calcium and then open the calcium channels, not as much calcium goes into the neuron. And conversely to that, increased magnesium, because magnesium and calcium tend to antagonize each other's function in the body in a similar way to the way in which sodium and potassium antagonize each other's function. The second class of block is competitive antagonism. A molecule comes along which attaches to the post-junctional receptor but doesn't stimulate that conformational change. It doesn't open the ion channel. So it's a competitive antagonist with the neurotransmitter. Classical examples there are the neuromuscular blocking agents that are used as anesthetic drugs, or rather used as drugs during the practice of anesthesia. They're not in themselves anesthetics, they cause paralysis. And the most commonly cited example of those, although there are many, is curare, which is the drug that South American Indians classically use to tip their arrows when they're going hunting monkeys. Hit the monkey with the arrow, monkey becomes paralyzed, monkey falls out of the tree, monkey becomes dinner. The next class are the direct depolarizing agents. Very similar to competitive antagonism, they get onto the post-junctional receptor, but this time they cause the receptor to undergo its conformation change and open its channel. And whoopee, you think, that leads to transmission of information across the synapse, and it does, but then those molecules sit on that receptor and force it to stay open. And it stays open, and so this post-junctional membrane stays depolarized, and it can only transmit the information once. So you tend to get an initial stimulation followed by a lack of stimulation. Classical example again, succinylcholine. It's another muscle relaxant drug used in anesthesia, and it causes muscle fasciculations, muscle contractions, followed by paralysis. Finally, we've got the indirect depolarizing synaptic blockers. These are drugs that block acetylcholinesterase. So remember I said that the acetylcholine in this synaptic cleft is cleaved into a choline molecule and an acetyl molecule, and it's cleaved by acetylcholinesterase, tissue acetylcholinesterase. These two drugs, well, this class of drugs really, and the example that I put there is the OPs, the organophosphates, but neostigmine is another one. There are lots of different examples. These agents block the enzyme that destroys the acetylcholine. And that means that the acetylcholine hangs around in the synaptic cleft longer than it should do. And so effectively, acetylcholine becomes a direct depolarizing muscle relaxant just because it's not metabolized and not taken away. So it has a prolonged activity. As I say, I'm not going to say anything more about dual block, and I'm not going to expect you to know anything about dual block. And that will do for today's lecture.